Hi, my name is Mariah Kidd, and today I'm going to be talking about positive inducible gene regulation in prokaryotes. So what does it mean to be positive inducible? Well, let's break this down. So to be positive means that we're going to have an activator protein present and something's going to start. To be inducible means that transcription is going to turn on. So now let's look at this and let's introduce our key players. So this is going to be my activator protein. As you can see from the curved edge at the top of its head, it's not going to be able to bind to our DNA strand here. However, when a small molecule or effector is present, the small molecule can bind, and what this does, it's going to change the shape of my activator protein. So now it's going to be able to bind to my activator binding site, which is just a sequence of DNA that codes for where the activator protein will bind. So now RNA polymerase is able to recognize my activator protein and transcription can happen. But I also need my promoter. The promoter is where RNA polymerase is going to bind to, and it knows to do this because it contains a certain DNA sequence. So if my small effector was not present, then the top part of my activator protein would never change and it wouldn't bind and transcription wouldn't happen because RNA polymerase wouldn't recognize it. So you would think that once it goes to the primer, transcription would just happen, but we have another step. There's an operator. So because we're looking at positive inducible, our operator is going to be turned on. The operator is kind of like an on-off switch for transcription. If you want to see an example where this is turned off, look at other examples of gene regulation. So finally, I'm going to have my structural genes. This is what's actually going to be transcribed, and it's going to then make other proteins. So now let's look at an analogy for this to make it a little easier to understand. So here we're going to have a young boy named Jimmy and his mom. So in order for Jimmy to do his homework, we need two things: a nagging mom and a stubborn Jimmy. So if we don't have a stubborn Jimmy, then the mom's not going to start nagging. But once we have the nagging mom, the homework's going to start to get done. So our nagging mom is going to be our activator. And since the mom won't start nagging unless we have a stubborn Jimmy present, he's going to be our effector or small molecule. And homework is going to represent transcription. I hope this helped you to understand positive inducible gene regulation in prokaryotes. Please check out these resources if you have any questions.